Hi, now we are going to see the endoplasmic reticulum, right? This is another important cellular organelle for your module test. Okay. So the definition or introduction is, it is the interconnected unfolded network. It is an interconnected unfolded network of tubular structures in the cytoplasm, right? Like a tubular structures in the cytoplasm. Actually, endoplasmic reticulum and Golgi apparatus, they are involved in the formation of lysosomes and peroxisomes. This is another function of this endoplasmic reticulum. Combined with the Golgi apparatus, they are involved in the formation of lysosomes and peroxisomes. And we have two types. In the school days also you studied about this endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum. Rough means involvement of the ribosomes, smooth means no ribosomes, like that. Okay. So a cytosolic portion of the endoplasmic reticulum studded with the ribosomes, which give a rough appearance of the endoplasmic reticulum, right? Whereas in smooth endoplasmic reticulum, it is devoid of ribosomes, that is, there is no ribosomal attachment. Functions of this rough endoplasmic reticulum is only one, that two very important functions: synthesis of protein. Okay. Whereas coming to smooth endoplasmic reticulum, there are two or three functions are there. The first one is synthesis of fatty acids, that is lipogenesis, synthesis of lipids. Second one is production of steroid hormones. Third one is, already we discussed, detoxification, that is toxic compounds are converted into non-toxic compounds and then excreted, that is drugs and toxins. So this is regarding the type of endoplasmic reticulum. Next important one, clinical point is, this endoplasmic reticulum involved in the protein misfolding and protein aggregation. Protein misfolding and protein aggregation that leads to formation of diabetes mellitus, cancer and Parkinson's disease. Very, very important clinical disorder: Parkinson's disease, diabetes mellitus, cancer-like diseases are involved in the protein misfolding and protein aggregation. This is the clinical problem associated with endoplasmic reticulum, right? Our next topic is Golgi apparatus or Golgi complex, right? So the introduction point is, it is a complex made up of a series of staked and flattened. These two words are very, very important. Series of staked and flattened pouches, small pouches. That is called a cisterna. What is the name? Cisterna. And vesicles. So it is made up of, actually it is a complex made up of series of staked and flattened pouches and vesicles. Where it is located? Next to the endoplasmic reticulum and near to the cell nucleus. Usually near the nucleus only it is situated. What is the function? Very, very important function. This Golgi apparatus is responsible for the transportation, packaging and modification of proteins. Three important functions they are doing: transporting protein and lipids, packaging into the vesicles, and modification, that is post-translational modifications of proteins and lipids, and delivers to the targeted destination, like act as a courier, right? Then the structure, actually in your question they have asked what are the compartments or describe the structures like that. For that the answer is, the Golgi apparatus has three compartments, right? One is cis compartment, another one is med compartments, third is trans compartment, right? Cis form, trans form, you already read in carbohydrate chemistry like that only. So cis form, med form, trans form. So what is the function of these three? Cis form is it receives the freshly synthesized proteins from the endoplasmic reticulum. The first organ which receives is Golgi apparatus, the freshly synthesized proteins from the endoplasmic reticulum. Second one is med. What is the med doing? Post-translational modification. Actually, they will be synthesizing in the inactive form all the proteins. After some reactions only, they will get active form, converted into active forms. That is known as post-translational modifications. In this med form only, this post-translational modification of proteins occurs. Third form is trans form. The name itself, it is stored, packaged into the vesicles and finally transported to the target organs. That is delivered to the targeted organs. So cis form, med form, trans form, all the three compartments you have to mention about the Golgi apparatus. Diagram is very important. For any cell organ question, you have to draw the cell diagram and mark a proper labeling.